Yes, hello, welcome.
Thank you very much for the introduction. Good evening, ladies and gentlemen.
You may use your full time. You're not restricted to 3 minutes.
Okay, thank you very much. So I'm Tsunapi. I'm representing the Urological Department of Totoro University from Japan. And I'm going to present about the nicotine-induced alterations in oxidative stress parameters in the rat bladder. I have nothing to disclose. So tobacco smoke, it is well known that it contains approximately 5,000 chemicals. It has also highly adverse effects in human health and also affects the urogenital system. Additionally, it induces generation of reactive oxygen species. Furthermore, cigarette smoking has been found to have a negative impact on continence, fertility, and aggravate symptoms of interstitial cystitis. So, taken all together, in the present study, we designed it in order to see the changes in the bladder that are induced by nicotine. We selected nicotine as the major addictive substance of tobacco smoke. And additionally, we checked the effects of abstinence in the bladder. So for our study, we used the 8-week-old male Wistar rats. So this means adult rats. So in the beginning of the study, we separate the animals into 3 groups. The control group, which was consisted of 10 animals and were treated with fresh water daily. The nicotine group, that also had the same number of animals and treated with nicotine at a concentration of 100 microgram per milliliter. The treatment was for 10 consecutive weeks. And we also had the abstinence group treated with nicotine at the same concentration for 7 weeks, followed by 3 weeks of abstinence. So here you may see the results and the general features of the animals. So 10 weeks of nicotine treatment induced a significant decrease on the body weight compared with the controls. Also, it induced a significant decrease on the bladder weight compared to the control, as well as the abstinence group. And also, we evaluated the ratio of the bladder weight to the body weight. And also, we saw a significant decrease in the nicotine group compared to the control. As a main metabolite of nicotine, we selected cotinine, and we evaluated the levels in the serum and the urine. So as you may see, both in the serum and the urine, the nicotine group had significantly higher levels compared to the abstinence group. As far as the histology now is concerned, 10 weeks of nicotine treatment induced some alterations in the histology of the bladder. As you may see, we can see the abruption of the lamina propria from the urothelium. Also, there was an edema and degeneration of the submucosa. In the abstinence group, we could not find this alteration. The pattern was closer to the control group. Furthermore, we selected malondialdehyde and 4-hydroxynonenal as markers of oxidative stress, of lipid peroxidation, and we evaluated by immunohistochemistry. So you may see that the expression of these parameters are strongly expressed in the urothelium of the nicotine group compared to the control and the abstinence group. As far as the 4-HNE is concerned, we could see a strong expression in the urothelium as well as in the muscle, smooth muscle area. And also we evaluated the 4-HOHD as a marker of DNA oxidative damage, and we could see a strong expression in the urothelium as well as in the smooth muscle area in the nicotine group. Because we didn't have the opportunity to perform functional studies, we selected the nerve growth factor and we checked the expression by immunohistochemistry because it is considered as a key protein for the bladder dysfunction, and we could see a mild expression in the urothelium in the nicotine group. So in conclusion, the nicotine-induced degeneration of the submucosa and edema in the substantial epithelium upregulated the expression of oxidative stress markers in the urothelium and the smooth muscle cells, and also induced an overexpression of the NGF, which is a key protein for the development of bladder dysfunction. So abstinence for 3 weeks could reverse the negative effects of the nicotine in most of the altered parameters and could restore the bladder histology. So the urologist may need to put effort to ensure that patients are aware of the relationship between smoking and urologic disease and motivate them to participate in a smoking cessation program as part of their treatment. Thank you very much.
Any questions from the floor? I assume nobody is surprised that smoking is bad for you. Taking that as a given, I have a question actually. There is increasing discussion that a lot of bladder pathology is secondary to changes in perfusion. And nicotine is known to affect blood vessel tone. So against that background, would you care to speculate whether the effects you observe are primary effects on the bladder or secondary to effects on the vasculature?
Probably it's kind of combination. Probably first happens. Yeah. Like smoking also induces an inflammatory response to the body. So it's more like all coming together, also affecting the bladder, not only the bladder, and leaves the rest out. So,
okay. I guess the last question of the night is for Mike.
Thank you, Mike, Jerry, Philadelphia. The mice didn't smoke; they got nicotine.
Yes.
So,
unfortunately,
you can make mice smoke. You have to put them in a chamber,
we know,
and give them
but
a cigarette, and blow it in. So there's a lot of things in cigarette that are, in addition to nicotine, that are really bad for you. Probably why you get cancer is not because of the nicotine, but for the other stuff that ends up in the bladder. So what do you think the effect of nicotine itself directly on the bladder muscle is? Has that been, have you studied that at all? Because I think it's been a very understudied field. I think everybody assumes there's no nicotinic receptors in bladder muscle, and I have data in my lab on dog bladder that that's not the case. Nicotine does contract the dog bladder. You can block it with atropine, but not alpha beta methylene ATP. It releases ATP. So I'm wondering if you think that may be partially mediating some of these effects you're seeing on the inflammatory.
Actually, yeah, thank you very much for your comment. As far as the first part of the comment concerns, unfortunately, we could not make the smoking experiment with the chamber because in our animal center, all the ventilation system is connected. So there was a problem with the other scientists' experiments. Yes. As far as the muscle area is concerned, it's a very important point. Yeah, we would like to see the contractions and see how is the reaction, but unfortunately, we did not.
Okay, Tony didn't understand the last question to Mike thing, but that's okay.
No, I just want to say one very quick thing. No, there are nicotinic receptors in the bladder. So there are intramural ganglia, but there's also nicotinic receptors in the urothelium. And that was shown by Bladder.
Thank you very much.
Okay, with that, I would like to close the session. I would like to thank you. I would like to thank all the presenters, discussion, and the audience, and of course my honorable co-chair, who asked me that he wanted to stay silent today. I wish you a great evening and see you back tomorrow.